Hello everyone. This is a presentation from Group 1 people from Commerce on protein expression and purification. The article given for us is on high-level expression and one-step purification of a soluble recombinant human interleukin 37B in Escherichia coli. Coming to the introduction, as the article mainly discusses about interleukin 37B, first we should know what are interleukins. Interleukins are a group of cytokines, which are nothing but secreted proteins and signaling molecules that were first seen to be expressed by white blood cells. The common families of interleukins include interleukin 1 to interleukin 13, interleukin 15, and interleukin 17. Interleukin 1 family consists of 11 members that are interleukin 1 to F1 to interleukin 1 F11. Interleukin 1 family 7 recently has been renamed as interleukin 37. Coming to interleukin 37, it has been found in monocytes, tonsil plasma cells, and breast carcinoma cells. Similar to other interleukin 1 homologues, interleukin 37B does not contain a typical signal peptide, but it possesses a potential proteolytic processing site for caspase 1 that could be necessary for its biological activity. The cleavage process of interleukin 37B precursor into a mature protein containing 198 amino acids mainly requires caspase 1. And the translocation process of mature interleukin 37B into the nucleus also requires caspase 1. Recent reports have showed that interleukin 37 inhibits several disorders such as renal, cerebral, and myocardial ischemia, and so on by suppressing the production of pro-inflammatory cytokines. In addition, interleukin 37 also mediates the anti-tumor activity in hepatocellular carcinoma, and it is also found as a natural suppressor of innate inflammatory and immune responses, but also inhibits adaptive immunity via induction of tolerogenic dendritic cells. The present study involves the expression of a recombinant human interleukin 37B in Escherichia coli at high levels as a soluble form and purified by one-step process called nickel nitrilotriacetic acid, which is abbreviated as NTA, agarose affinity chromatography. Coming to the methods that are involved in protein expression and purification process. This process mainly involves plasmid construction, expression of recombinant interleukin 37B, preparation of bacterial cell lysate, which is nothing but E. coli lysate in this process, purification of recombinant interleukin 37B, STS page analysis and protein quantification, followed by Western blot technique and N-terminal sequencing, and finally activity assay. Let's have a look at each step in detail. Coming to the plasmid construction. In this process, the cDNA, called complementary DNA, was amplified using polymerase chain reaction, which is abbreviated as PCR process. The amplification profile included one initial hot start denaturation step at 98 degrees centigrade for 2 minutes, which is followed by 30 cycles of the following conditions such as 98 degrees centigrade for 10 seconds, 62 degrees centigrade for 15 seconds, and 72 degrees centigrade for 60 seconds. And a final extension was made at 72 degrees centigrade for 10 minutes. And the products that are obtained from PCR process were purified through agarose gel electrophoresis, and the gel band containing desired materials were purified through QIA quick gel extraction kit. A recombinant plasmid, which is designated as PET44 interleukin 37B, was constructed by inserting the amplified fragments into the restriction sites of PET44. And finally, the transformed materials were verified through enzyme digestion and DNA sequencing. By this plasmid construction process, the final protein was able to express the recombinant interleukin 37B and containing the hexahistidine tag, which can be purified through nickel affinity chromatography. Expression of recombinant interleukin 37B. The successfully transformed E. coli was picked up from a single colony and was grown overnight at 37 degrees centigrade. The culture mixture was provided with fresh medium containing ampicillin and was grown with continuous shaking. Once the sample had obtained absorbance of 0.5 to 0.6 at 600 nanometers, the interleukin 37 expression was induced by the addition of 1 millimolar IPTG, which is nothing but isopropyl beta-D-thiogalactopyranoside. And then the incubation was continued at 37 degrees centigrade for periods of 1, 2, 3, 4, 5, 6, and 20 hours. And finally, the STS page analysis was used to determine the degree of expression for the various time intervals. Here we can see the pictorial representation of STS page analysis of recombinant interleukin 37B protein expressed by E. coli. As I said before, the expression was induced by 1 millimolar IPTG at 37 degrees centigrade for different periods. Here lane M represents the protein molecular weight marker, which was used as a control, and lane 1 represents the whole cell lysates before IPTG induction where there was no protein expression. Whereas lanes 2 to 8 represents whole cell lysates after 1, 2, 3, 4, 5, 6 and 20 hours of IPTG induction at 37 degrees centigrade respectively, where there is a production of a fusion protein of approximately 22 kilodaltons, which was well matched with its theoretical molecular weight. As we can see here, the results showed that while the incubation period was below 6 hours, the yield of the 22 kilodaltons fusion protein was increased as the induction time prolonged. But when the incubation time exceeded 4 hours, the yield of the product was not significantly raised. Preparation of bacterial cell lysate. The bacterial cell lysate was obtained from a 6-hour incubated sample. After 6 hours of induction at 37 degrees centigrade, the medium was cooled down and the cells were harvested through centrifugation process at 4000 grams for 20 minutes at a temperature of 4 degrees centigrade. The supernatant was discarded and the pellet was stored at minus 20 degrees centigrade overnight, and the stored pellet was then suspended in 8 ml of phosphate buffer saline using vertexing or pipetting up and down. The cells were disintegrated by an enzyme called lysozyme for 30 minutes at room temperature with continuous shaking. Sonication on ice was also used to agitate the sample until it was no longer viscous, and also the bulk cellular fragments were removed with centrifugation at 12,000 grams for 30 minutes at 4 degrees centigrade. The supernatant was collected and was syringed through a 0.45 micrometer sterile filter membrane to remove the remaining particles. Purification of recombinant interleukin 37B. The filtered supernatant was mixed with nickel nitrilo triacetic acid agarose, and the mixture was agitated with continuous shaking at 4 degrees centigrade for 1 hour. And then the mixture was loaded onto a column, which was washed with a wash buffer of pH 8, and the interleukin 37B protein was eluted using an elution buffer with the same pH of 8, and the protein was desalted and concentrated by ultrafiltration process and was stored at minus 80 degrees centigrade for later use. STS page analysis and protein quantification. The purified expressed protein was resuspended in STS page loading buffer and fractionated after being denatured. The proteins were then stained with 0.1% co-massy brilliant blue solution, and the electrophoresis was performed with mini protein 3 electrophoresis cell. The protein concentration was determined through co-massive plus protein assay kit, and the protein purity was estimated using densitometry of the protein bands from images of the STS page gels. And from the results of densitometry, the purity of the protein was found to be greater than 97%. Western blot technique further identified the specific interleukin 37B protein and the N-terminal sequencing confirmed that the first 10 residues were identical to the interleukin 37B. Here we can see the complete process that is involved in the purification and expression of a protein. As I said before, lane M represents the markers which was used as a control, and lane 1 represents the uninduced cells where there was no expression of protein, whereas lane 2 represents total cell lysates where we can see the expression of interleukin 37B protein. And lane 3 represents the pellet which was used for the preparation of a bacterial cell lysate, and lane 4 represents the supernatant which was used for the purification process. Whereas lanes 5 to 8 represents the purified interleukin 37 protein which was analyzed by STS page analysis and protein quantification. And lane 9 represents the Western blot for uninduced cell lysate, and lane 10 represents the target interleukin 37B protein which was appeared as a single band and further confirmed by Western blot technique. Coming to the activity essay, here there is a graphical representation of interleukin 37B protein effect on the production of tumor necrosis factor alpha and interleukin 1 beta in lipopolysaccharide stimulated THP1 cells which were incubated with 5 to 500 nanogram per mils of recombinant interleukin 37B for about 24 hours. As we can see here, the results have shown that the recombinant interleukin 37B decreased significantly the release of pro-inflammatory cytokines that are tumor necrosis factor and interleukin 1 beta in lipopolysaccharide stimulated THP1 cells in a dose-dependent manner. And also the tumor necrosis factor alpha and interleukin 1 beta concentrations in the culture medium were determined by using ELISA kit, which is called as enzyme linked immunosorbent assay kit. Coming to the conclusion, this study led to the isolation of recombinant interleukin 37B protein in one step using immobilized metal affinity chromatography. This procedure also gave interleukin 37B of high purity and high activity. And this method is well suitable for the industrial production and will be significantly beneficial for further structural and functional studies of interleukin 37.